Hi there, this is Mrs. Ortega, and I am back with you here. We're going to look through chapter 33, which will be invertebrates. So any animal without a backbone. This chapter is a little bit long, but we will go through and look at all the exciting animals that are in the group of invertebrates. Invertebrates are any animal that will lack a backbone. They do not have that nice vertebrae column that we do have in our backs. And so they account for more than 95% known animal species, which is a very large amount. And they are also morphologically diverse and occupy almost every habitat on Earth. And so for this, essentially knowing what invertebrate is where we want to start. And so looking at a phylogenetic tree, we saw in last time as well, animals all have an ancestral protist that leads to a common ancestor of all animals. And from there, the first group to branch out is Porifera, which is the sponge group. And so that's where we will start today. And as we continue along, we'll head down this chain and to get to the other more complex groups of the Cnidarians here, and then the other groups, the Lophotrochozoans, Ectoprocts, and Deuterostomians we saw in that very end of chapter 32. And so the next few pages here show you the group along with its species, like the phylum name, the amount of species in that group. And so of these, we'll only look at a few, or we won't look at all of them. That's pretty much impossible. But we'll look at those two here, Lophotrochozoans. We'll see actually these four here, all four of these here. And more Lophotrochozoans. We'll see these here. Ectoprocts, I believe just these two. Sorry. Deuterostomians. We'll look at the echinoderms, and then the chordates, which will lead to the vertebrates. But the hemichordal that we mentioned at the very end of last chapter is the acorn worm, and that's shown there. So if you're wondering what that looks like, that's what it looks like. There are 85 species of acorn worms. All right, so we'll start off with the first group, the Porifera, the sponges. These are the basal animals that will lack the true tissue. And most of these are going to not have symmetry as well, or the most common group does not have symmetry. Some of them do. And so this group is known informally as sponges, but their technical name is Porifera. And I guess from the pores are in their tissue, or lack of true tissue structures. So sponges are sedentary, so they're not mobile, and for most of their life they will live in one region of marine or fresh water. They're known for being filter feeders, which is a key trait here. So as we go through these, we'll look for key traits and distinguishing features of these groups for you to know. I'll try to make sure I point everything out as well. One of them being filter feeders. They have unique cells that will capture food particles as they filter that water through their body. And we'll see that in the next slide here. Another key feature is that they lack true tissues and organs. And so that's something we'll see. And so the water, as it filter feeds the water, is going to go into the sponge through openings and it's going to go to the central region called the spongocoel, which if you remember from last chapter, the ending seal means cavity, so sponge cavity. It's hopefully making sense in terms of naming here. And then the opening that the water will leave is called the osculum. And so you can see that in this next picture here. So this is a great slide to look at, and that the water flows in, and then the pores that are labeled pores here that allow water to flow in are actually called ostia. You'll see that in labs. So I like to kind of correlate names together. Ostia are the pores that allow water to flow in. Then the water will flow into the center region, which is the center cavity, the spongocoel, which is here. And so that's this region here. And then it's going to leave through the osculum. And so that's how they filter the water. The actual process of capturing the food is done by a cell called the choanocyte. So that's up here. The choanocyte is very similar in structure to the protist we mentioned last chapter, which is the choanoflagellate. This is a protist. It's a choanoflagellate. This is the protist. Oh, I skipped ahead. Flagellate. Sorry. This is a protist that looks very similar in structure to this choanocyte, or also known as collar cell, for this distinct feature of projections that are in a circle, and this is called a collar. These projections trap the food particles in nice slimy mucus, and the reason or the way these food particles move onto this collar is through the flagellum. It's going to propel it like a jet for a boat. It's going to propel it into a circle, and that will capture food particles more easily. And then from there on, for that mucus will move the food particles to this body of the cell, where they will phagocytosis or ingest the food particles. From there, those phagocytosis vesicles will move through the choanocyte cell to what's called the amoebocyte cell. The amoebocyte cell will be responsible for distributing the food to all cells in the sponge, as well as some other functions. And so that's generally, we'll just, I believe, stick with that. They're kind of the food distributors here. Also, you'll see some supporting structures. These spicules are these crystal projections or spikes that help in protection as support for the sponge. And then the mesohyal, which is a gelatinous tissue that will kind of hold everything together. So I believe that's it from this line. And so some definitions for you if you like words and terms. Choanocytes are flagellated collar cells. They generate the water current through the sponge and ingest suspended food. And so the amoebocytes are going to be next to those choanocytes, and they will play roles in digestion and structure. So they're going to be breaking apart that food, spreading those nutrients through all the cells, and structure as well. They'll fill in some regions. The mesohyal will be a gelatinous non-cellular layer between the two cell layers, the inner and outer layer of the sponge. And then unique facts about the sponge are that they are hermaphrodites. Each individual functions both as a male and a female. Typically, the way that sponges reproduce will be that sponges will start off as male. They'll function as male, release sperm through the water, and then that has to find a female sponge that release eggs throughout the water. And so from there, that will then form the embryo sponge, and that will actually be mobile until it finds a place to settle down and grow into a dormant sponge. And so that sponge functions first as a male and then can adapt or eventually become a female. And so that's a unique trait of sponges. And so here, wants to focus on listing our review of choanocytes, osculum, and spongocoel. So choanocytes, also amoebocytes, and then the spongocoel and osculum. Alrighty. Moving right along to our next group of animals, the Cnidarians are going to be an ancient phylum of eumetozoans, so that's true tissues. These are animals with true tissues, but they will be very different than other animals in that they have a diploblastic radial body plan, which is not typical. So they are having just two of those germ layers of endoderm and ectoderm, and they have radial symmetry. And so this is actually a wide range of animals, including jellyfish, coral, and hydra. And so those are some that we'll look at here. A unique feature that they all have in some form or fashion is a central digestive compartment known as gastrovascular cavity. This gastrovascular cavity will have one single opening that functions as both the mouth and the anus. And given its term here, gastro meaning like our gastric system where we break down nutrients, break down large particles of food into smaller particles of food for nutrients for the cell. So that's half of what it does. The other half is a vascular portion that will be responsible for the acquisition of oxygen along with the release of carbon dioxide. These animals will use the gastrovascular cavity for both nutrition, digestion, and as a circulatory system will circulate the food to all nutrients to all cells and collect waste from all cells. So that's the unique structure we want to understand. And so let's take a look at the Cnidarians here and their unique body plan, which is radial for both. But they actually have two different body plans. There's a sessile plan and a motile or mobile plan. So the sessile plan is a polyp. This will be stationary. It's not going to adhere to the substrate by one end of its body, whereas the medusa is going to be mobile and going to move. And so that's a key difference here. And you'll want to understand these two body plans. And so they move, the medusa move freely. And so looking at those two plans, you can see they've color coordinated the different germ layers and what they develop into. So here you can see on the polyp, the stationary, I like to think that the tentacles are face up. So they face upwards. That's also a common feature both body plans have for the Cnidarians will be tentacles. So around in the center of the ring of tentacles will be the gastrovascular opening, so that's the mouth or anus. And so inside there you can see the nice big gastrovascular cavity for the polyp. And then the endoderm is actually, in yellow there is forming what they call the gastrodermis, or the dermal layer of the gastrovascular cavity. And so looking at polyp, it attaches to a substrate on this end, gastrovascular cavity, and then mouth and tentacles face up. Compare that to your medusa form here that has a more bell-shaped curve to its body on the top, And then the tentacles are facing downwards on the bottom, along with the opening for their gastrovascular cavity. And so they move using their tentacles freely throughout the marine environment. And so this is something you want to know. Looking at their tentacles, they use them to capture prey. They also use them in defense when they feel threatened. And if you've been stung by a jellyfish, you know it's not pleasant. But these tentacles are armed with specialized cells that will function in that stinging sensation that will help with defense and capturing prey. And so the cnidocytes are these specialized cells. In these specialized cells, they have organelles that are specialized for this stinging, and those are called nematocysts. So those, for us, you want to focus on the cnidocyte, the cell itself, versus the organelle. And so you can see that in this next image here. So this is an example of a hydra. So on their tentacles, they have multiple cells, as you see along the tentacle, that will then go, if you look closer up, inside the cell, there is the nematocyst, which has the coiled up thread that can discharge and insert into the skin of the prey or predator, if you will, that's wanting to defend from. And so that will cause a stinging sensation. It can also inject poison, just depending on the group of species, into that other organism. And so for us, the cnidocyte is a stinging cell. That's what you want to focus on in terms of this portion here. In terms of classifications, the cnidarians have two major groups, the medusozoans and anthozoans. And this diverged early in evolutionary history. So you can see they're very different in terms of structure and appearance. And so if you were to guess that the anthozoans are mostly they're divided mostly because the anthozoans are polyp form and medusozoans are medusa form, you would be correct. And so looking down here, medusozoans are all cnidarians that produce a medusa body form. And so subgroupings are cyphozoans, which are known as jellies, cubosocins, which are box jellies, hydrozoans. And so in hydrozoans, from there you have the hydra, which is actually only in polyp form, that has unique reproductive capabilities. And so looking at hydrozoans, they usually alternate between polyp and medusa forms. And so those, I think you would usually see in lab class as obelia is one of them. The other name is, can't remember the other name at the moment, but if I think of it, I'll tell you. And so here, I have a video of a jellyfish for you. And so this, you can see how it moves. It's really cool. It kind of moves by pushing its tentacles downwards or bringing them upwards and along the edges, however you picture it there. Right, and then for Hydra, I have a picture of one and then a video as well. So we'll look at the Hydra. Usually in lab class, we actually have some live ones. You can see them under a stereoscope, the light brown, large microscope looking thing. But unfortunately, you probably won't get to see that live this semester, thanks to COVID-19. But here you can see the Hydra has the basal disc that attaches it to a substrate, which for us, they can freely float in water, as you would see if you got to see them live, but they can also attach to like the edges of the container as well. And then moving, you can see they're nice stalk or that will have the gastrovascular cavity inside, and then on the ends they have the tentacles that surround the mouth or a hypostome region. The hydra is unique in that it's only in polyp form, but it does have a asexual reproduction and sexual reproduction. As you can see, they are also hermaphrodites. They have both gamete reproductive organs, and then they can grow asexually, which is shown here. And so it's very unique, very interesting. Most people might be familiar with the name Hydra as that seven-headed monster from mythology, but that's not the case here. It's actually a very, very tiny, very small little cnidarian. And so I'm going to play that video for you of it eating another small organism called the Daphnia. And so that is unique or cool to see. So it captured it with its tentacles, probably stinging it repetitively until it calms down. And now it's going to start swallowing it. It's almost done swallowing, it looks like. Okay, it successfully has swallowed that thing whole. And so now it's going to work to break down those parts in that gastrovascular cavity. Will release enzymes to break it all down, which is nice. All right, moving right along here. This is the Obelia example of a hydrozoan. And so in this case, it alternates between a polyp state and medusa state. And so in lab, I believe you would see the polyp state only, and that under a slide, you would see this lovely structure, usually dyed like a pinkish color. And so looking at the polyp forms, there's a feeding polyp version that has tentacles, and that reproductive polyp that will eventually form a medusa that kind of buds out. And so that medusa there will then produce have the gonads to produce the gametes, the egg and the sperm, that will then travel by water, fertilize, form the zygote that will then develop to the larva. The larva will then develop into a polyp that will then mature. And so it's a nice alteration of generations from polyp to medusa, but for cnidarians, not for plants. So keeping some terms here for you. The anthozoans, they are going to be known for their stationary form, the polyp form, and they include the corals and the sea anemones. They are known also for their hard exoskeleton, which is made from calcium carbonate, something you've seen before. And so when carbon dioxide goes into the ocean, it reduces the amount of carbonate that's available for calcium to bind with. And so that will in turn affect the coral reefs. So the amount of carbon dioxide in the atmosphere, in the oceans, is actually really bad for coral reefs. And that process you might have learned in Bio 1 is called ocean acidification. When carbon dioxide enters the ocean, it has very bad effects. Sorry, I don't have a great writing ability on here. And so each coral reef, when it grows, it will form, when it lives, and then eventually it will die, and that those minerals will decay and actually form hard rocks that provide habitats for other species in the next generation. And so the generations build upon the skeletal remains of previous generations. Alrighty, so let's see here. For those two, in terms of groupings, before I move on, I want just to make sure, keeping the just general ideas here, that anthozoans are mostly polyps. The medusozoans have the medusa. So that's what you want to focus on there. All right, moving to the Lophotrochozoans. They have a wide range of body forms, and they're identified by molecular data. And so that's more recent. The bilateral animals in this group have bilateral symmetry and triploblastic development, with a coelom and digestive tract with 2 openings. And so the clade bilateral includes those 3 groups of Lophotrochozoans, ectodizosans, and deuterostomians. Just keep that in mind. It's not hugely important for us to know those arrangements, but it does help in terms of understanding. And so Lophotrochozoans include flatworms, rotifers, ectoprocts, brachiopods, mollusks, and annelids. And so we will look at these animal groups. And so looking at them, we'll see in some of them they'll have the lophophore structure for feeding. Others will have a trochophore larval stage. And then some of them won't have either of these features. So that's how they're grouped together, is based on those developmental features that we saw at the end of chapter 32. And so looking first at flatworms, the phylum name is Platyhelminthes. They live in either marine, freshwater, or damp terrestrial habitats. They have a triploblastic development, so remember those 3 layers, ecto, endo, mesoderm. But they are acoelomates. They don't have that true body cavity. But they do have a gastrovascular cavity with 1 opening. And so I think that should be one listed on here. Actually, it's not listed on your review, but it is something that you should know, that they have that nice gastrovascular cavity, but in terms of their vascular portion, it's mostly for circulation. Their gas exchange is going to take place on their skin. And then they have something called protonephria that regulate the osmotic balance. So you can kind of think of like a premature urinary system for the flatworms. And so their flattened shape is dorsal to ventral surface. So top to bottom or bottom to top. And this maximizes the surface area needed for gas exchange. And so if you look here on this example of maximizing surface area, you might be familiar with something like this from BIOL 1 when you look at cell size. They keep a small cell size to maximize the surface to volume ratio. And so looking at this image here, they have the same volume of 27 cubic centimeters, but one of them, the flatter one, has the higher surface area. And so that's to help you understand why these flatworms are so flat. And so in terms of maximizing surface area, the flattening can occur, which is shown here. Branching can occur, like in the mycelium or hyphae for fungi. Projections we'll see in later chapters of the cilia, projections, and microvilli projections in our intestinal system. And then folding can occur, which we saw in plants with the thylakoids and the chloroplasts. So, all right, more about flatworms. They have 2 lineages, one of which we'll focus on, the Rhabdatophora. Sorry, circle correctly. This is the one we'll look at, but there is a second one there called the Cannulida, or chainworms. But the best known rhabdatophorans are the planarians. They live in freshwater and prey on smaller animals. But they're known mostly for being free living species, meaning that they're not going to be parasites. And so other flatworms we'll see are parasitic. And so overall, though, their structure, they will have light sensitive eye spot regions, centralized nerve nets for premature nervous system, and they are also hermaphrodites and can reproduce sexually or asexually through fission. So looking at their body plan, you can see here the Dugesia is a planarian that you might look at in lab. If I can write, I'm sorry. That looks very similar to this drawing here. And so looking at the mouth is an opening on the ventral surface, that's the bottom surface, the dorsal is the top surface. And so the mouth is an opening, and out from the mouth comes the pharynx, which is a tube-like region. It's going to suck food in. Then the gastrovascular cavities, and this dark brown here that is throughout the whole body of the flatworm. It has a left and a right side as you move forward, further down there. And then just like the left and right of the gastrovascular cavity, the nerve cords will run ventrally, and so those are left and right as well, and those lead to ganglia that are also with the eye spots at the front. The Dugesia itself has something called an auricle, which is a region that helps detect like tactile reception, and so that's something else kind of along the edges here. But for us, this is the flatworm. It's non-parasitic. The planarian flatworm is non-parasitic. All right, so on to the parasites. Parasitic rhabdatophorans live in or on other animals. And so two important groups of this are the trematodes and the tapeworms. In the trematodes, you'll see in your lab class, hopefully, these are flukes. So flukes like the blood fluke is one that comes from contaminated water that when ingested can cause many different things, but the most notable one, the blood fluke, has a male and female flukes that live together in unison, and so that allows them to reproduce efficiently in large numbers of eggs that will leave through the feces, I believe, that will then possibly contaminate more. But we'll focus on the tapeworm here in lecture. Tapeworm is going to lack a digestive system, as they are going to absorb nutrients from their host directly through their skin. And so looking at the tapeworm itself, the structure is very different than its non-parasitic relative, in that it has a head region or anterior region called the scolex that will have both suckers and hooks that will help attach it to the host. Usually in the intestine, when the tapeworm is at maturity, it will be living in the intestinal system. And it will produce units of the tapeworm will be in different units, and these units are called proglottids. They will contain both sex organs for producing egg and sperm, and so these will attach behind the scolex, and eventually after the eggs are developed, the sperm is developed, then it will become fertilized. and then eventually detach at the end of the tapeworm and leave the host body through feces. From there, ideally for the tapeworm, other animals will ingest the contaminated feces, like pork, pigs are known for ingesting that contaminating feces, and then will develop larvae in their muscles. And so that's how humans will then get the larva through eating undercooked pork. And from there it will go, the larva will develop into mature adults in the intestinal system. And so as we can see here in this picture, the tapeworm region, the interior region with hooks and suckers for attachment, and then the long units of proglottids one after another with reproductive structures. And so the beginning ones will start developing the eggs and the sperm, and which will be mature. At the end, they're going to be ripe because they're going to be fertilized eggs ready for growth into larvae. And so while they are very dangerous and can be very detrimental to human health, I've actually heard of some doctors, especially in Central America, that will use these as a weight loss tool, which is highly frowned upon now. All right, so I think that wraps up our worms, the flatworm group. Looking at the next group are the rotifers. Rotifers are like Phylum Rotifera. They are very tiny animals that will inhabit freshwater, ocean, and damp soil. And they're actually smaller than many protists that we've seen in chapter 28. But they are actually multicellular and have specialized organ systems. As you can see in this image here, they're known for their crown of cilia around their mouth, and then from their mouth goes to their jaws, where they have a complete digestive system of stomach, intestines, and then eventually anus. What's also unique about these rotifers is that they are can be completely see-through, which is very cool, along with their tiny size. Some species are actually known for being completely female only, that they lack males entirely, and can reproduce from unfertilized eggs. They also have a unique function, some of them that we'll call cryptobiosis, where they can undergo a dormant state, where they basically just stop living, and then they can resume living at some point, depending on different factors. But for us, rotifers are smaller than many protists, but are multicellular. So that's a key fact you want to know from the previous slide here. And then that they have females that reproduce offspring from unfertilized eggs. So that's not necessarily written on here, but starting, I guess, with this portion, females only, and they reproduce from unfertilized eggs. So that's the rotifers. Moving to the ectoprocts and brachiopods, these are the Lophotrochozoans, are known for the lophophore, which is the crown of ciliated tentacles around their mouth. We saw that in the rotifers as well, but for ectoprocts and brachiopods, they're further divided based on their resemblance to other things. So the ectoprocts resemble plants because they are sessile colonial animals, of which they have a hard exoskeleton to protect the colony. They are also known for helping build the coral reefs. whereas the brachiopods will resemble clams and other mollusks, but their shell is their two halves of their shell are dorsal and ventral, so top and bottom, rather than right and left in terms of orientation. And so they are going to usually be sessile in that they will attach to the sea floor by a stalk. And so you can see that in this next slide here. Along with their key differences, that will be what you'll want to know. Ectoprocts have a hard exoskeleton, so they don't have a shell. They're sessile colonial animals that have the lophophore structure for feeding, whereas the brachiopods are attached by a stalk and have a dorsal ventral shell, just so it's shown there. And so you can think top and bottom hinge, and so that's their orientation as to where they're sitting is top and bottom, where we'll see in the mollusks their shells are left and right, or lateral. Which is next. The mollusk group, they have a lot more in terms of diversity, so they're further divided into other groups. And so they include snails and slugs, oysters and clams, octopuses and squid. Most of them are marine, some of them inhabit freshwater, and some are terrestrial. But they're all gathered by the fact that they are soft-body animals that are either protected by a hard shell or have a hard shell remnants. And we'll see what that means in different groups here. In terms of their body plan, they have three main parts, which is, I believe, yes, something I want to know, so keep that in mind. Their muscular foot is one that can be used for movement. It depends on which one you're looking at, but usually the muscular foot is pretty pronounced. They have something called a visceral mass and a mantle. And so looking at these, it's best to see the general image of a mollusk here, and that their foot is usually on the ventral surface, a bottom surface that's used for movement. And as you can see, it is innervated, or has nerve cords running through it that help in terms of detection. And then the second portion, the mantle. The mantle will produce the shell, which is right above that. And then the visceral mass is pretty much everything else. So this will have the soft, squishy body that contains all of the organs responsible for life. And so you can see a complete digestive tract running from the mouth, has a radula, a claw-like feature that drags food in, all the way from there to the stomach, intestine, and then eventually anus. They have gills for gas exchange that will lead to the body cavity, the coelom, and the heart there. And so that's going to actually have a open circulatory system for the most part, where their organs will kind of bathe in this fluid that will circulate throughout the body. But overall, three main structures: muscular foot, visceral mass, and mantle. In terms of reproduction, they have separate sexes with different gonads in the visceral mass, but many snails are hermaphrodites as well, being able to produce both there. Some mollusks will have a ciliated larval stage that corresponds to the trochophore that we saw, a larval stage in the Lophotrochozoan groups that helps connect them to the rest of the animals in this group. But in terms of mollusks, we'll want to know their three clades, which is nicely depicted here, of gastropods, bivalves, and cephalopods. And so for gastropods, they are going to be snails and slugs. So some are terrestrial, most are marine. Their slugs have lost the mantle and the shell, so they only have 2 of the 3 corresponding features that all mollusks have. So that's a bit confusing, but let's look at those pictures here. About 3/4 of all living species of mollusks are gastropods. So here you can see snail and sea slug. Those slugs and snails. You just want a video if I can have one in motion. And so those sea slugs are pretty elaborate in terms of their appearance and projections that hopefully allow for them to camouflage better in those bright coral reef regions. Alrighty. And so here describes gastropods a little bit more, that they are moved by rippling motion of that foot or by cilia projections. Some that have a shell will have a single spiral shell that will function in protection from injury, dehydration, and predators. Most of them are herbivores, but some of them will use that radula to feed on prey. All right, so moving to the second group, the bivalves. Bivalves will have shells divided into 2 and hinged together, and this will be laterally from left to right. And they're known for being sedentary suspension feeders. And so that's what you'll see here. But they include clams, oysters, mussels, and scallops. They are their halves of the shell are drawn together by adductor muscles. They will have sensory tentacles or eyes along the edge of their mantle. So remember, their mantle produces their shell, so that'll be kind of on the interior edge of the shell will have some sensory organs. So here's another one example. So the mantle cavity of that bivalve will have gills that are used for feeding and gas exchange. Most of them are sedentary. Some of them have limited mobility. And so the last group here, the cephalopods, which literally translates to head to foot. So cephalo meaning head, pod meaning foot. Gastro, you can probably assume means like stomach, foot. And so cephalopods here will use beak-like jaws to rip or crush prey, and they have large brains and elaborate sense organs. So looking at those, we have here, before I show you the cephalopods, the last tidbit of the bivalves are here. I'm sorry, didn't remember how picture of this. Just the overall organization of their internal structures, along with their just different regions there. And so that's very, very good for if you're studying bivalves in particular, but for us, this is a little bit extra information. But if it helps you understand them better, then great. But looking at cephalopods here, they are going to be carnivores, so they're going to be eating other smaller organisms in the ocean. Usually in the marine environment, they have beak-like jaws, and these are usually not very apparent because they are surrounded by their tentacles. So their tentacles usually surround their mouth, and so inside of their mouth will be that beak-like jaw. And so in lab class, at least when I've taught it, I will have them dissect a squid. And so you can see that in the squid. Hopefully you get to see that in some sort of form of dissection that you might do. And so they have many different unique adaptations, like immobilizing prey with poison. They can creep along the seafloor in search of prey. And so here are some common examples, the squid being one of them. The squid has a very apparent ink sac that it can inject out of its siphon. Siphon fluid is just like what you think it would be spelled like. The siphon will help it with water, with gametes, egg and sperm, fertilization, and then for their ink will all come out of this tube-like region called a siphon. I believe the squid actually only reproduce once, at least for males, and so they obviously don't do that often. Because I think they actually will die after reproducing. So here next we have the octopus. Octopus is known for its very large tentacles, its very elaborate complex eye structure, and then its brain. Some octopus can actually camouflage as well, so it's very elaborate in terms of sense organs. And so they also have their mouth at the bottom, like the ventral side surface, that will have a beak-like thing to crush and rip prey. Then you have lastly, the chambered nautilus is one of the few that still has its shell. As you can see here, the squid and the octopus do not have external shells. The squid will actually have something called a pin, which sounds weird, but a pin will actually be in the internal portion to help stabilize the structures of the squid. That runs kind of up and down the squid right there. And so that's like the remnant of the shell. The octopus, I'm sure, has one as well. I just am not super familiar with that one. But the chambered nautilus will have the external shell, and that's one of the few that do still. And so here it describes more about this group. Squids, their siphon will let out water, ink, and gametes. Cephalopods will have a closed circulatory system, so they're going to have veins and arteries that will allow for transport of their blood to and from their heart, gills, and brain, other body parts as well. And so the one group of shelled cephalopods and nautiluses are the only group that survives today. And so I'm going to check here. Just due to timing, I'm going to go ahead and stop this recording, and we'll have a part 2 for this chapter, just for those that want to kind of have some organization. And so on the next video, I will pick up here with annelids. So bye-bye for now.